and that is awkward. Regarding gene expression and regulation. Also, by the end of this, should be familiar with some common laboratory techniques and hopefully some more cutting edge ones as well. Also, another goal is to be able to understand scientific literature in the field of molecular biology, and also be able to interpret data from techniques that we're going to cover in class. So if you were to go into any scientific article and look at the data figures, you would be able to understand what they're trying to convey. This is the course schedule. So today is going to be pretty basic. We're just going to go over macromolecules, so DNA, RNA, and proteins, and their structure and function. Next week, we'll be going into genome structure and integrity. So that will include things like DNA packaging, DNA repair, replication, and we'll be going into some techniques as well, such as DNA isolation, PCR, which is something that's been of interest lately because of COVID testing. And session 3 will be gene expression. Well, 3, 4, and 5 will all be gene expression. First, we'll be going into transcription, which is the process by which DNA is read and transcribed into RNA. Then for that session, we'll be focusing more on RNA. The session after that, we'll be focusing on translation. So that's gene expression 2. So the translation is the process by which RNA is translated to amino acids to create proteins. And then the last gene expression session will be on protein regulation and how proteins are controlled in the cell to yield a certain trait or phenotype. And then from session 6 onward, we'll be focusing on how genes are regulated. So we'll be covering things like epigenetics and non-coding RNAs. I think for session 7, we may have a guest speaker who is more knowledgeable about gene regulation than me. So that's still TBD, but I'll let you know when I get that confirmed. And the last session will be split between model organisms. So those are organisms that are used in laboratory settings to study gene regulation that are analogous to humans. And the last section of the last session will be hot topics. So for hot topics, that is really up to the class. So if there are any suggestions for hot topics, anything you're particularly interested in, you can just shout that out right now. So if anyone has any ideas, just unmute yourself and
how about CRISPR?
Anyone else?
Oh, maybe about the mRNA vaccines. I know that's a hot topic.
Okay.
Real quick, I noticed that there was an article about the complete genome being, human genome being done. And I was interested in, that was the 23 chromosome pairs, and now they're supposed to go to the 46. And I was interested in that. Also interested in, there was 2 people that had a genetic defect, and they were blind, and they have tried a gene splicing thing to try to fix that. And I believe that, at least one of them I read was successful, that they could see again. So I was interested in finding out, if anybody has current topics of how they're applying genetics and genes, like the messenger RNA and all. Thank you.
Anyone else want to chime in? Okay, if you have any ideas that come up later, feel free to put those in the chat, and I'll include that in my notes for when we have the last session. So, I think by now I'd be accustomed to Zoom, but still. Okay, so gene expression is based on a concept known as the central dogma of molecular biology, which describes the flow of genetic information. And this, in simple terms, is the flow of information from DNA to RNA through transcription. In this case, we're talking about messenger RNA or mRNA, and then from RNA to protein through translation. In reality, it's a lot more complicated than this. In addition to transcription, we also have reverse transcription, where RNA can be reverse transcribed into cDNA or coding DNA. In addition, DNA maintains itself through replication. And this is a schematic of how that occurs with the molecular machinery involved. So transcription takes place through the use of enzyme called RNA polymerase, which binds to a coding strand of DNA and then reads it to yield pre-mRNA. The pre-mRNA is then processed to yield mRNA, which is then translated into a polypeptide or protein through the use of ribosomes with another form of RNA called tRNA or transfer RNA. So we'll be going into more detail about that later, but this is just a visual schematic of how the central dogma takes place. So before we get into the biology, we need to also establish some basic vocabulary in terms of chemistry. So the macromolecules that we'll be talking about today, DNA, RNA, and proteins, they're all comprised of subunits, which form a macromolecule. So in order to form these macromolecules, certain reactions have to occur. So some vocabulary you should be familiar with before we go into this are hydrolysis, which is cleavage of chemical bonds by adding water, dehydration synthesis, which is the forming of polymer bonds by removing water. And these reactions are important in modulating the secondary structure of biological molecules. This will be especially important when we're talking about proteins, where the structure is affected by the side chains of the amino acids, which we'll be talking about in more detail later. In addition, the bonds between the monomers are also important in terms of macromolecule structure and function. So you're probably familiar with several of these already. So the bonds that will be important include ionic and covalent bonds. Ionic bonds are the ones where an electron is given from one element to another. A covalent bond has shared electron pairs, and then we have phosphodiester peptide and hydrogen bonds. Phosphodiester bonds are going to be relevant when we're talking about DNA in particular, and peptide bonds are going to come into play when we're talking about proteins. In addition, hydrogen bonds will be important in both of them. So first, we're going to talk about nucleic acids, which includes, which in this case, we'll be talking about DNA and RNA. So before we get into the structure and function of nucleic acids, I just want to have like a brief discussion of what you already know about the structure and function of DNA and RNA, especially DNA, since a lot of that is common knowledge. So, yeah, feel free to just unmute yourself and throw out what you know about DNA structure or function of both DNA and RNA.
Sorry, Shirley, you're asking for people to speak up about, to say.
Yeah, just what we know just from what their basic knowledge is of structure and function of either DNA or RNA.
Okay.
Yeah, Jonathan?
Sure. Let's see. DNA deoxyribose nucleic acid is a double helix structure, whereas RNA is a single structure, like it's usually like a strand. So I'm trying to go over all the basics that I remember. But yes, so they both have nucleotides, which are their like monomer components. And let's see, DNA, it's adenine I remember it by the letters A, C, G, T, and RNA is different because instead of instead of the T nucleotide, it has the uracil, which funny, I can remember that name, but yes. So yeah, that runs the gambit of my most of my knowledge so far. Yeah.
Anyone else? You keep going. Okay. Okay, so in terms of function, DNA and RNA have distinct functions. So DNA's main function is to store genetic information. So because it's a storage molecule, it is stable. So in a lab setting, if you have DNA, you leave it on at room temperature for like a month, it's fine. Whereas with RNA, it's extremely unstable, and that's because it has a short-term function. It's involved in gene expression primarily. So there are three major types of RNA that we'll be discussing in this class. There are also ways of using RNA to manipulate cells, such as silencing RNA or RNA interference. So that will come in later as well. But in terms of RNA that's present in the cell, the main ones are messenger RNA or mRNA, which we'll be talking about today, transfer RNA or tRNA, which we'll briefly talk about today. This is involved in the translation process, and ribosomal RNA or RNA. We won't really be covering this one. So yeah, as Jonathan was saying, there are some structural differences between RNA and DNA. The first structural difference I want to talk about is the sugar structures. So the two sugars involved in DNA and RNA are deoxyribose sugar, which is in DNA, and ribose sugar, which is in RNA. The main difference between the two types of sugar are at the 2' position. So in DNA, the 2' position of the sugar molecule has a hydrogen, whereas in RNA, it has a hydroxyl group at that site. As Jonathan also mentioned, another major difference between DNA and RNA is that DNA is double-stranded, RNA is single-stranded. Both are comprised of nitrogenous bases, which are represented here, and they also contain a phosphosugar backbone, which is formed through this high-energy triphosphate structure, which is shown here.
Shirley, I have a question. You know, I'm familiar with the 2', 3', 5', but I don't know what's the system for numbering. Like, how do they decide to go one way versus the other way in terms of numbering the carbon atoms?
Yeah, I'm not really sure. Beth, do you know by any chance?
I think it's just kind of the tradition in organic chemistry to start with the carbon that's closest to the most functional base. So in this, or the most functional atom. So in this case, it starts at the O, and the next carbon in the ring is number 1. The one after that is 2. The one after that is 3. The one after that is 4.
Yeah.
And so they just, it's kind of tradition, I think, in organic chemistry to number that way.
And you go clockwise.
Yeah. I don't know why though.
But it depends on which side you're looking at it from, whether it's clockwise or counterclockwise.
Yeah.
It's just always like I say, how do they know it's, you know, like
the
orientation? Okay. The minor point is just kind of like hit me like, well, why?
You're gonna have to ask the organic chemists.
Yes.
I make a point of avoiding those people. So when the monomers are bonded to create the macromolecule, there's a directionality to it. So so each monomer has what's called a 5' end and a 3' end. So this image represents DNA, but the same concept applies to RNA. So this triphosphate group is the 5' end, and this hydroxyl group is the 3' end. And usually when you're talking about DNA in a lab setting, you're referring to it in the 5' to 3' direction. And so that's just the convention. I'm going to focus a little more on DNA now. So DNA is a double strand, as I mentioned. It has complementary pairs of that are comprised of pairs of four nitrogenous bases. Those bases are adenine, thymine, guanine, and cytosine. So thymine pairs with adenine, and guanine always pairs with cytosine. And you'll notice that the number of bonds between each pair is different. So thymine and adenine only have 2 points where they're bound, whereas guanine and cytosine are bound at 3 points. This detail is important because it affects the temperature. It affects how the DNA molecule behaves at different temperatures, which is especially important in techniques such as PCR, where you have to melt the DNA to separate the 2 strands. If you have a higher percentage of guanine and cytosine, you're going to need a higher temperature to separate those strands. Whereas if you have a lot of adenine and thymine, that DNA molecule is going to melt at a lower temperature. And the larger, the macro structure that's formed by the 2 DNA strands is this double helix, which consists of a major group and a minor group. And this is just.
Could you say like what does it mean, major groove and minor groove?
Yeah, so basically there are 2 gaps that are formed between the two strands. So the larger gap is the major group, the minor group is the smaller gap.
And this is relevant because there's a lot of proteins that like DNA, and some of them will only bind in certain like orientation. Some of them like the major groove, some of them like the minor groove, and that they interact in very specific like ways. So we use that language quite a lot when we're talking about protein DNA interactions.
Okay, so when it's in a double helix form, the 2 complementary strands are they're in opposite directions. So, for instance, this bottom strand is going from the 5' to 3' direction. The complementary strand is going from 3' to 5'. So in this case, you have one strand that's referred to as the coding strand, and the other one is referred to as a template strand, which is important during transcription because when the mRNA is formed, it's formed based on the coding strand, not the template strand. So I think we have some time for a break now, but before we do that, I just want to share a simulation of DNA structure. I know we allotted time for about a 15-minute break, so I think what we can do is I can send that link in the chat, and the simulation only takes a couple minutes to go through, so maybe everyone can do that independently and then take a 15-minute break, and then we can come back at 7. Does that sound good? Okay. Okay, send a chat box.
Okay, there we go.
So the next macromolecule we'll be talking about is proteins, which are comprised of amino acids.
My bad. Ignore that.
Okay. Okay, so the process of getting from DNA to RNA is called transcription. So just rewinding a bit. So again, just wanted to remind you, there's this coding strand and a template strand. So the enzyme RNA polymerase recognizes the coding strand and yields this mRNA. There's mRNA processing in between, but we'll get into detail with that later. So now that you have this mRNA and you need to get this, you need to translate this so that you have a protein sequence. So this is carried out in the ribosome, and here the tRNA or transfer RNA is used to recognize the mRNA sequence, and from there it's translated into a sequence of amino acids or polypeptide. So the way that mRNA is read is through codons. So those are sets of three nucleotides. So this is this wheel here is the genetic code. So each combination of three nucleotides will yield a certain amino acid, which is listed on the outer rim here. So there, you'll notice that there are multiple combinations of nucleotides that can yield one specific amino acid. So there's 20 in total. In addition, there are also codons that code for stop, which designates where in the sequence the ribosome should stop producing the polypeptide. In addition, there's also a start codon, which is the methionine. So at the beginning of every sequence, every coding sequence will be an AUG, which codes for methionine, and that is the start codon for that polypeptide. So this is a visual of how that takes place. So then this takes place in the ribosome, and you have the transfer RNA or tRNA, which is complementary to a specific codon, and is conjugated to an amino acid that's specific to that codon. So the transfer RNA will recognize a codon in the mRNA, and it will bring the amino acid to the mRNA, and then there will be a peptide bond formed between the amino acids, leading to a growing peptide chain. So amino acids all consist of an amine group and a carboxyl group. So these are also referred to as N and C. In addition, there is a side chain, which is designated here by the R. So much like how in DNA and RNA you read from 5' to 3', in amino acid sequences you read it from the N terminus to the C terminal direction. So from the amine group to the carboxyl group. So as you before, there are 21 amino acids, and the difference between all of these is the R group. So that is what the R group is, a side chain, which is represented basically anything that's between the amine group and the carboxyl group. So there's a lot of variation here, and here they have it categorized into 5 groups. So you have arginine and lysine, which are positively charged, and then aspartic acid and glutamate acid, which are negatively charged. Then you have amino acids with uncharged side chains that are polar. So these include serine, threonine, asparagine, and glutamine. Then you have amino acids with hydrophobic side chains, which are all listed here: alanine, valine, isoleucine, leucine, methionine, phenylalanine, tyrosine, and tryptophan. And lastly, you have these special cases. So those include cysteine, which can form disulfide bonds due to the sulfur here. That's going to come in later when we talk about techniques that are used in terms of protein isolation or such as and protein analysis, such as Western blot. There's also selenocysteine, which is a variant of cysteine. Then glycine, which is the smallest one. This affects how, because of its size, it affects how the protein is folded. And the last special case amino acid is proline. So unlike the others, this one has the amine group in a ring, which also affects the secondary structure of the protein. And the reason why the characteristics of these side chains are important is because they affect the secondary structure as well as the function. So for instance, if you have a stretch of hydrophilic amino acids, that stretch of the protein is able to enter membranes, which are lipid, which in the cell are made of lipids. So those are also hydrophobic. If it's hydrophilic, then it can it's more likely to be in the cytosol. In addition, it also affects protein folding. So if you have a protein that's in the cytosol, but it has a hydrophobic stretch, it can fold in a way that the hydrophobic stretch will be in the interior of the protein. Those are all aspects of protein regulation that are affected by the amino acid structure.
Shirley,
yep.
I have a very basic question. Why, this is for everybody, why is it called transcription? And why is it called translation?
Because you're transcribing from one nucleic acid sequence to another, but then you're translating from one type of molecule to, at least that's how we were taught, how I was taught to remember it. I don't know if that's the actual reason for it. But yeah, when you're translating, you're going from one type of molecule to another, whereas when you're transcribing, you're still working with nucleic acids.
Oh, I think of it as because the code AGCT is really the same. DNA and RNA, AGCU, it's actually just T without the methyl group on it, right?
Yeah.
Or with the methyl group, can't remember which way around it goes. But so it's just copying. You're just copying from one molecule to the other. Then when you're creating an amino acid polypeptide chain, it's a translation because now you have 20 different amino acids. So I think that's how I kind of remembered it.
Okay. And this is again something that we'll go into more detail in a later session. But protein structure has multiple levels. So on the most basic level of a protein structure is the primary structure, which is just the amino acid sequence. So that's one amino acid after another that are joined by a peptide bond, and it has a distinct amino end and a carboxyl end. From here, it can form a secondary structure. So some examples of secondary structures are the alpha helix or the beta pleated sheet, and which form the secondary structure is going to take is dependent on the hydrogen bonds between amino acids at various locations within the polypeptide chain. In addition, it can also be affected by cysteine residues through disulfide bonds. There are also higher levels of protein structure called tertiary structure, which is the globular protein structure that you've probably seen in papers. And then there's quaternary structure, which is only occurs in certain proteins that have multiple subunits. So those are levels of protein structure. So now I think we have time for another break, and then when you come back, can go over any questions that you have, and then we're going to end with an interactive activity. So I think we can take is 5 minutes, okay? Okay, so yeah, let's come back at 7:07.